All right, welcome everyone. In this video, I'm going to tell you about my expectations for scientific writing in microbiology BIO203, specifically about the research paper you're going to write that goes along with the unknown lab practical. So you all have this same rubric. It's attached to the rubric I gave you in class on the day that we started the unknown lab. And I just want to walk you through it. So all scientific papers are broken down into four major sections. There's an introduction, a methods, a results, and a discussion section. And then they're all followed up with a references or a works cited page. Your paper will follow this same format. So make sure that when you lay out your paper, you include these headings: introduction, methods, results, and discussion. And then lastly, your works cited. I've got it broken down in each section, the information that you need to include. So as long as you have information that explains each of these bullet points, you should have all the content that you need to in your paper. So really, this rubric is more like a template that you can use to fill in all the information you'll need in your paper. All right, so in your introduction, the first thing you'll do is introduce your patient. What were their signs and symptoms? Next, you'll discuss the general steps on how you identified the unknown bacteria and why or how the methods are important for the identification process. So for your gram stain, you did the gram stain to figure out what steps you had to do next in your testing, your biochemical tests, right? And also which antibiotics to put on your Kirby-Bauer test. So it's all about how you did these things and how they helped inform your identification of the unknown organism. He'll talk about the cell morphology and arrangement, the biochemical tests. You'll discuss how the above methods led you to identify your unknown organism. So you won't actually include any of the methods for your biochemical testing in this paper. Keep that in mind, please. You'll just report what you like, the results of your catalase test and how that helped you to understand what your unknown is. And then this is where you'll state the identity of your unknown bacteria. So keep in mind that the paper you're writing is not about identifying your unknown. It's more about your patient. So it's about identifying what's making your patient sick and then figuring out which antibiotic will be effective in treating your patient. All right, and then the last bit here for the general steps will be, was there a key test or two that let you know what the organism was right away? And then discuss that. Next, you'll talk about describing your unknown organism. So you'll begin with that cell morphology and colony morphology. You must include the following information in this order. We need to know your gram stain reaction, the cell morphology and cell arrangement, the cell structures. So how did this look? Did you see like any flagella or now that you know what it is, you can research that organism, right? So you're going to read about how it uses structures like pili, flagella, maybe a capsule, or maybe it's an endospore former. You're going to have to look this information up and use in-text citations. Colony morphology will also be discussed here. And then you're briefly going to discuss the 5 antibiotics that you chose to treat your patient and why. You must include some brief background information on the mode of action for your antibiotics and why they should be or could be used to treat this infection caused by your bacterium. All right, this is where you'll state your general hypothesis and objective. So that might be something like you hypothesize that, the penicillins will be effective in treating your patient or something. The last sentence of your introduction should explain, why are you writing this paper or doing this experiment? Remember, the whole goal of this paper is to figure out how to save your patient, right? So keep that in mind. Something else is that you don't need to report your results in the introduction. So you don't need to tell us which antibiotic was the most effective. You're just going to tell us about the antibiotics that you're testing. Okay. All right. Your methods section. And you can actually begin writing your methods section after day 1 of the unknown lab practical in the lab, because we're only discussing the methods for the Kirby-Bauer test. That's lab 11, the antibiotic sensitivity test. All right, so you're going to tell us what you used and how you did it. So describe how you performed the Kirby-Bauer method. This is lab 11. You can paraphrase it. Be specific. The reader should know exactly what you did and how you did it so that they could repeat the experiment using your instructions in the methods. You should also include in the methods section, and you won't be able to do this until after the second day of lab, but you should also include how you analyzed your Kirby-Bauer test. How did you determine sensitivity or resistance? All right, the results section, this is where you explain what did you discover. So in paragraph form, you'll describe the results for each of your 5 antibiotics. Was the bacterium susceptible, resistant, or maybe intermediate? And you must include your data as a table as well and properly reference it in the text. When you reference a table in the text, it'll look something like this. You describe the results in text, but then you reference where they can also be found in a table. Does that make sense? All right, the 4th section, your discussion. This is where you tell the reader what you think your results mean. And especially keep in mind that your reader might be your patient, or it might also be the public or other practitioners. So you're going to discuss the significance of the unknown organism, your bacterium, the pathogen. You're going to include statistics on the number of cases each year in the US or worldwide. What are the reservoirs of infection for your organism? And what disease can that organism cause? Next, you're going to talk about treatment. Which antibiotic or antibiotics did you discover that could be used to treat this infection? If your results did not yield results for effective treatment, what does the literature, what do other scientific studies say is most commonly used or most effective against your organism? You're going to discuss your results. Was your bacterium resistant to any of the antibiotics? You're going to research your organism and consider the different types of infection your organism can cause. Are all infections treated the same way? Is antibiotic treatment even recommended? Why? Last, you're going to talk about prevention. Discuss ways infections caused by your organism could be prevented. Include specifics for your patient scenario on how this disease could have been prevented. And then last, you're going to have your references or your works cited page. I've already given you a sample of what a works cited page would look like on your APA formatting guide. And you can use this to format your reference page. You should have a minimum of 3 credible resources, and your textbook should be one of these because your Kirby-Bauer procedure comes from your textbook, right? So you can reference your textbook here in your methods where you describe the Kirby-Bauer procedure. Something to keep in mind is that it is not appropriate to take direct quotes in scientific writing. So you're going to paraphrase the procedure. You're not going to copy and paste it. All right? Okay, so keeping this in mind, a good paper should be about 6 pages double-spaced, and you're going to use APA formatting for your works cited page and for your in-text references. So wherever you say something that someone who's an intelligent but not scientist person, so someone who's not a microbiologist wouldn't know it, you need to provide a citation for it, okay? All right, so 6 pages, double-spaced, like regular 12-point fonts. I'm not going to be super nitpicky on your fonts, but just so you know what I'm looking for. If you're using too big of a font or too small of a font. Your length might be different, but you, in order to meet all these points here in the rubric, it's going to take you about 6 pages. Does that make sense? So I'm not necessarily counting pages, I'm looking for content, but a good one usually ends up being about 6 pages long. All right, let's move on. There's a second document I want to talk to you about. And that is what makes good scientific writing. So actually, I'm going to include that in a second video. So we'll pause here for now. And of course, if you have any questions, please let me know.